Dr. Mojarrad is an infectious disease physician. He's a scientist, and he's globally renowned expert in vaccinology, virology, immunology, and emerging pathogens. He started at my brother's university, Duke University, and then he got his MD, PhD, Masters of Public Health through a National Institutes of Health funded National Medical Scientist Training Program at University of Alabama, Birmingham. But then he has a litany of really great universities that he interfaced with for his fellowships and his residency, starting at Yale, Vanderbilt, and then at the National Institutes of Allergy and Infectious Disease in Washington, D.C., and he had a distinguished career there in Washington. Most recently, for the Military Health Institute's perspective, Dr. Mojarrad is the founding director of the Emerging Infectious Disease branch at the Walter Reed Army Institute of Research in Washington, D.C. Okay? And then he was very active over the last 2 years in the Operation Warp Speed program at the executive level. More importantly than that even, he was detailed, if you will, to the World Health Organization to help with the global vaccine response. So he's got a wealth of experience. He's had a really full 2 days already with our university and also the UTSA colleagues in the immunology department there. And it's been a very active dialogue today, and we're looking forward to your comments tonight, Kayvon. Welcome.
The title of my talk is a little bit different than what's posted outside and on the advertisements. And I did that intentionally because I thought, well, I'm being hosted by the Military Health Institute, and previously I was my institute, Walter Reed Army Institute of Research, that I just recently left, was housed within the Army Futures Command. And one of the things that we got repeated and impressed into our heads is that we need to think about the future wars and not fight the wars of the past. In my world, that means thinking ahead in terms of what are the next pandemics, the next epidemics that we have to be on guard for. And one of the ways that we can anticipate and counter these pandemics in advance is to develop new vaccines that are broader, that are longer lasting in how they protect individuals, and are flexible enough that they can be turned on a dime to whatever pathogen might come our way. As we've seen over the past couple of years, that it's not just COVID out there. There are plenty of other viral pathogens in general, but take monkeypox, RSV, and all these things in the news now, and that's just the tip of the iceberg. So before I go any further, I have a few disclosures. The first is I'm going to be talking to you a little bit about a ferritin-based vaccine. I'll explain what that is, that I'm an inventor on for the patent. The data I'm going to be presenting, the little bit of data that I'll be presenting, is completely in the public domain from work that I've done previously as an employee of the U.S. Department of Defense. But as of 3 weeks ago, I am now an employee of Pfizer. And I won't be talking about anything related to Pfizer products. And so because of that, I think that I've done my due diligence to mitigate all potential conflicts. So we're going to start with a pop quiz. And I want you to be honest when I ask you to raise your hand in response. You don't have to blurt out the answer, just raise your hand. So how many of you recognize who these two people are? Okay, most of you. And I'm not being flippant here, okay? I'm going to come back to this. Now, how many of you recognize who these two people are? You got to recognize both of them. Wow, okay. I'm going to come back to you, sir, at the end and see what your responses are, but that's very impressive. So before I move on, yesterday as I was settling into the hotel, I turned on the TV and put it on public television, PBS, and there was a documentary about the Justinian plague in ancient Rome. And so a lot of you know about the bubonic plague and the Black Death in the 1300s, but there was a huge plague in Constantinople, and a lot of the remnants of the Byzantine Roman Empire back about 800 years before the Black Death. And it was focusing on, I didn't even know these existed, but sort of viral archaeologists who now we've developed enough technology to be able to go into the skeletons of people of deceased 1,500 years ago and extract their teeth and bore into their teeth, and there's still dried blood in those teeth, and then sequence out viruses that may, or bacteria, or any other pathogens that may have been circulating in the blood. And doing so, they were able to definitively confirm the pathogen that was causing this plague. It was Yersinia pestis, exactly the same one, and actually they were able to draw out the genealogy that it was related to the same plague that caused the Black Death 800 years later. But they were also able to confirm that this plague lasted for over 200 years, close to 250 years. And at a time when historians have thought that the fall of the Roman Empire, it was sacked in 412 AD, but then about 150 years later is when this plague occurred, and Rome and the Byzantine Empire was still pretty robust, still pretty dominant, and people thought it was militarily, they became weak, and they had a number of battles that they lost, and that's eventually what led to the ultimate demise of the Roman Empire. When in fact, they think that it was this protracted plague coupled with a lot of climate change events at that time. Sounds familiar? That those two conspired to really weaken that empire. And so I bring this up because having been in a military institute doing research on viral pathogens, we constantly had to make a justification and case for how what we did was relevant to the U.S. military and the overall mission of the U.S. military. And I think history has always borne out that these other factors that are causing death and destruction just outside of the battlefield really are some of the most major factors that we have to be concerned about. And so at the end of this talk, I want to come back to this and talk about what has been the role of the military in biomedical research, particularly in the countering of emerging pathogens, and what should it be going forward. I've been talking a lot with a lot of folks in the past day about arcs, the arcs of people's careers, their research, and sort of the arc of everything that's been happening over the past 3 years or so, and what the arc of the pandemic is going to be over the next few years. And so I wanted to first talk about the arc of a mission. So, as I mentioned, infectious diseases have played an important role in history, but particularly in military history as well. So much so that a lot of the majority of casualties have been due to diseases and not combat-related casualties, to the point where it was called the Third Army during the Civil War. And born out of the Civil War in that period in the late 19th century, there was a strong focus within the Army of developing the infrastructure and the knowledge and the capabilities within the Army to study and develop countermeasures against this Third Army, inclusive of infectious diseases. And so here's a picture of George Sternberg and the first Army Medical School founded in 1893. That is the forerunner of what eventually became the Walter Reed Army Institute of Research. It is debated whether this was the first school of public health in the country. It goes back and forth between Johns Hopkins and the Army. and it depends on how you define a school of public health. But very early on in the history of infectious disease research in the military, there were some very important discoveries made, most notably by the namesake of the institute at which I worked, Walter Reed himself. And Walter Reed really rose to prominence by studying mosquitoes. And that's one of the reasons in that first slide I have a big mosquito hovering over the world, because mosquitoes, we can say viruses, but I think mosquitoes have rival viruses for killing more people than any other organism that ever existed. And in terms of the pathogens they carry. And until Walter Reed came around and work that he did with Carlos Finlay in Cuba, no one really understood that mosquitoes could carry these pathogens. That then directly informed the efforts of William Gorgas to go down into Panama and essentially eradicate yellow fever so that people could actually build the Panama Canal. And the Panama Canal really ushered in a new age, dominance, regional dominance and global dominance for the US as a whole. So infectious diseases have always had a really important role in global history and particularly in US history as well. So the next arc that I want to touch on is the arc of an idea that has been born out of the recent epidemics and pandemics over the last 10 years. If you're talking about H1N1, go back further to H5N1 and SARS-1, and then more recently the Ebola outbreak in West Africa, and then Central Africa, the Zika outbreak in the Western Hemisphere, and then most recently the COVID pandemic. The idea is that we keep encountering these new pathogens emerging from all corners of the world, and it's become somewhat overwhelming. How do we constantly respond to these without exhausting our resources, and respond in a way that is smarter, faster, more efficient, and more preemptive? So with the Ebola outbreak in West Africa, things started to change. Rather than folks saying, well, we had another outbreak, another epidemic, it's over now, let's go back to business as usual. After the Ebola outbreak in 2014 in West Africa, where a disease of case fatality of 90% moved from small villages, small isolated villages in Central Africa to some of the largest cities and most densely populated cities in the world, people started to finally notice that these things were not just going to stay contained in remote corners of the world. Even so that it came to our shores and came to Texas as well, right? So there was a lot of successes in the response, but there were also some shortcomings that people noticed. One of the things that was noticed was that we had a vaccine for Ebola on the shelves for about 15 years, 10 to 15 years before the West African Ebola outbreak. How come there wasn't a vaccine ready to go and be deployed immediately? That was because it had been in monkeys, but it had never been in humans. So they had to do all the clinical testing in real time as the epidemic is unfolding. We were lucky enough to be able to deploy that vaccine, one of those vaccines, get some efficacy data, and have it impact some people's lives, but not before close to 12,000 people perished in that outbreak, and not before other interventions actually made a greater difference in terms of behavioral interventions. So people started thinking about, well, we've known about Ebola for about 40 years. We started our vaccine program really in earnest around the turn of the century. And then finally we got to a licensed vaccine about 15 years after that. If we think about and I've just took a few examples of vaccines that have been developed over the past century, from the time that a virus has been discovered to the time that a vaccine was made, that timeline actually hasn't shortened at all. In some cases, like Zika, it's probably even longer. But from the time that a vaccine program has been started to the time that a vaccine has been authorized or approved, that timeline has shortened considerably. But with Zika, and I'll point out in the next slide, we still don't have a vaccine. SARS-CoV-2, obviously everybody knows the story. We got to a vaccine from identification of the pathogen, the first sequences, to an authorized vaccine in less than 1 year. So faster than we ever had before. I guess the question that I want to ask is that fast enough? So if you look at Ebola on the top, these are the epidemic curves of Ebola and Zika on the left side. In Ebola, we have the case counts in each of the primarily affected countries of Guinea, Liberia, and Sierra Leone. And you can see this is the first cases. This is when the public health emergency of international concern is declared by the WHO. And all the way here is when the vaccine trials start, the efficacy trials. Way after the peak. So all the behavioral interventions that really had the most impact had already started by the time that we got here. Still, despite the lateness of these trials, the guinea ring vaccination study was able to give us some efficacy data. It's really even worse for Zika because by the time the public health emergency of international concern was declared by the WHO, that was February 1st of 2016, right around that time, vaccine programs had already were started as well. That was also past the peak. And why it's worse is that we never were able to get efficacy trials completed to lead to licensure of a vaccine. So, next time Zika comes around, and it will, and it already is popping up in pockets of the world, we won't have a vaccine ready to deploy immediately. We'll have to test it in some capacity as we're trying to roll it out. And then COVID. So this is as of December of last year, so it's not even counting the number of deaths and cases over the course of this year. And these are reported COVID-19 deaths, so not the excess mortality. That is here. The estimates are about 15 million in terms of overall excess mortality in addition to the directly confirmed deaths due to COVID. And so having a vaccine within a year didn't really matter much for these folks. And it's not to take away from the amazing success that occurred with the development and the authorization of these vaccines. It's only to point out that we have to constantly push ourselves to do better. That we have to have these vaccines already on the docket, ready to be deployed to be able to contain outbreaks and save potentially millions of lives even in those first few months of the emergence of a new pandemic. So if the human cost is not enough, then one thing that I learned in the federal government as well, and we do this in academia and we do this in industry, is that we have to make the financial argument as well. That one for COVID was a pretty easy one, and one that we didn't expect. The largest estimates were for a pandemic flu costing about $4 trillion. So far, until this year, it's cost about $18 trillion to the global economy and will continue to do so. And so each of these outbreaks was making the case more and more. At what threshold was it going to catch people's attention in terms of stakeholders and funders and policymakers? I hope that we've reached that threshold by now. So what do we do? When we make these arguments of the human toll and the economic costs of all these different epidemics, how do we prepare for and respond to all of them? We can't. There are hundreds, thousands of viruses that can cause disease in humans. Well, we can focus on a number that cause major pathology, disease, and mortality, but we can even focus it even more into classifications of maybe about 24 viruses, virus families, I should say. And so that's what a number of different organizations, led by a organization that was born out of the World Health Organization's review of itself in the aftermath of the Ebola epidemic, this organization called CEPI, the Coalition for Epidemic Preparedness Innovations. That's when they decided we need to start investing in the development of vaccines in advance of the emergence of these pandemics. And they did this initially by coming up with a list of priority pathogens that had pandemic potential. And you can see here, coronaviruses were on the list. At the time, it was Middle East Respiratory Syndrome, which it only caused, in relative terms, few cases and deaths compared to a lot of other pathogens, but still, because it was a coronavirus, there was concern that it or a relative of it would have the potential to cause a pandemic, which it in the end did. Disease X became SARS-CoV-2. So there are ways that we can try to focus on prototypical pathogens within a handful of families of viruses and develop certain technologies that will allow us to anticipate more than one pathogen at a time. And I'm going to be talking about that a little bit later in the talk. So we have a general sense of what's coming. If we look at the discovery of new viruses over the past 100 years since viruses were first discovered, we see an exponential growth of virus species that are being identified. If we then categorize those viruses that are being discovered into their phylogenetic families, it really starts to plateau at a couple of dozen virus families. So we are able to start getting a handle on these. And there's no more poignant example than coronaviruses. SARS in 2002, MERS in 2012, and then here, Newsweek, with the start of the pandemic, wake-up call. It begs the question, why were we sleeping, why were we dreaming this whole time? Why did we need a wake-up call? Because we should have been anticipating something like this, maybe not the scale, but yet another coronavirus, because there were two other milder forms of human coronaviruses discovered in the interim between these two. So hopefully the world has now woken up to the possibility that this is not going to be the last coronavirus, this is not going to be the last pandemic. So then the last part I wanted to talk about is the arc, or the second to last arc, is the arc of an invention. And here I have in small letters, intervention, because that's what you hope an invention will become, at least when we're talking about vaccines, that it develops into something that can intervene to mitigate disease and save lives. Key point that I think we tend to lose sight of is that despite everything we've learned over the past 2 years about coronaviruses, it is still a relatively unknown family. Yes, there have been a number of different coronaviruses before this, but there are long periods of time initially between the discovery of coronaviruses at all in chickens to discovery of coronaviruses in humans, 40 years, and then from human coronavirus discovery to the emergence of the first highly pathogenic coronavirus, SARS, there was another 40 years. Now that timeline is starting to compress somewhat. If you look at the number of publications on coronaviruses this doesn't even go further back than the mid-90s, because there was just maybe a couple dozen papers before that. 98% of publications on coronaviruses have been since 2002. 90% of coronavirus publications have been since 2020. So we've learned a lot, but there is quite a bit to go in learning about this family of viruses. And particularly what we did not know and what we underestimated was the evolutionary potential of these viruses. Despite it being a negative sense RNA virus, we thought still it was probably a genetically stable virus. Probably wasn't going to be as bad as something like flu. Well, nature always has ways of surprising us. If you look at the time-resolved phylogenetic trees for influenza A and influenza B over the course of about 40 years or so, you can see the diversity of the viruses continues to expand. There was a really brilliant scientist in our group at the Walter Reed Autoimmune Institute of Research, Morgane Roland, phylogeneticist, evolutionary biologist, who initially in the early days of the pandemic showed a comparison of SARS-CoV-2 phylogenetic tree. And then basically this is the family tree of viruses, and compared it to flu and HIV, and it was just a little dot compared to this huge dendrogram of different species and strains for the other viruses. That has changed quite a bit in the ensuing 2 years. So the strategy that has been taken and has been very successful for coronavirus vaccines was based on technology you could draw the line in multiple ways. You could say it was based on technology from 10 years prior in the context of structural biologic resolution of the analogous protein to the spike protein of coronavirus in respiratory syncytial virus, called the F protein, or you could draw it back further to HIV and flu. The point is that without the investments that were made 10 years before, 40 years before, we wouldn't have the vaccines that we have today. So when people say, oh, we got vaccines within a year, it's really a year plus 10, year plus 20, year plus 40. And it was all enabled by a revolution in structural biology to be able to develop the right antigens. There was also a revolution in the platforms and how these antigens were delivered. I won't go into the whole story of mRNA right now, but I'll talk instead about a platform that we think is promising, or we thought was promising at Rare, to be able to get ahead of these viruses in such a way that we can elicit broad immune responses that not only cover the virus right there in front of you, but future ones, they may emerge in the future. And we did this based on a platform, a protein called ferritin. Ferritin is in pretty much all living things. It carries iron in humans, in bacteria, in plants. And so we used a bacterial ferritin that had been used in the context of a universal influenza vaccine program at the NIH, where I had been before, and my co-inventor on the vaccine, Gordon Joyce, had been before, and saw a lot of promise in preclinical studies that this will potentially give you a cross-reactive immunity to viruses in different groups of influenza. What makes ferritin so attractive as a scaffold, if you say, for the presentation of things like the spike protein is that it has a very nice geometry and symmetry that allows the presentation of proteins that are essentially triplets. And a lot of the surface proteins that mediate the fusion of the virus to your lung cells or other cells, they exist in this triplet formation. And so ferritin allows you to keep that native conformation. The other attractive feature about this platform was that just before the pandemic, it had gone into humans for the first time in the context of flu. So what we did is we took the spike protein that was stabilized in a certain shape that was based on work that was done with RSV that a lot of the major and most successful vaccines used, developed at the NIH, coupled it with another component, another chemical called an adjuvant, which helps boost the immune response. And that was also from the Army as well, called Army liposomal formulation Q for a compound called QS21. And that had also gone into humans just before in the context of a malaria trial. And we came up with this combination, this mixture of particles and these globules that represents the adjuvant. It's important that they had gone into humans because we could move more quickly with the FDA to be able to reference back to those studies and allow us to get into the clinic to evaluate in humans more efficiently. So what does the ferritin actually look like? So as I said, what it does is it starts as just a single unit, but on its own it polymerizes and forms this 24-faced cage. A lot of interviews that I've discussed this, I refer to it as a soccer ball. So it's spherical, but it has these different faces on it. And on these faces, you can attach whatever you want. In this case, that tripleted spike protein. You can use that same spike protein over and over again around the face of this soccer ball, or you can use different spike proteins so that you have kind of a mosaic of different particles being presented. What we went through in those first 2 months, and a number of you will remember this who were there at the time and leading our institute and our command, that we were all hands on deck, like everywhere, and to get moving as quickly as possible. And we evaluated about over 200 candidates and about 2,000 mice in the course of 2 months to come up with the best candidate to take forward into larger animal studies and into humans. We still did do a lot of work with other kind of top candidates, and these may prove to be valuable for future designs for what we call universal vaccines, but I'm going to focus on this one, which we ended up calling spiffin, for spike ferritin nanoparticle. And what does spiffin do? So I'll run quickly through this. What we started with was just the single spike protein. And the single spike protein will interact with the antibodies on B cells, or the receptors on B cells, and make them produce a bunch of antibodies against that single spike. But if you have different spikes on the same nanoparticle, all these different B cells can come interact with that single particle and start pumping out all different kinds of antibodies against different variants or different species of viruses. You can do that on a single particle, or you can make a cocktail of particles and still get the same result. The idea ultimately then is you get all these different antibodies that pre-exist in your body so that whatever virus you're presented with, if it was exposed to the particular spike or portion of the spike of that particular virus, you're already protected. And so we went through a lot of different animal studies, tested these in different strains of mice, different types of monkeys, hamsters, horses, nurse sharks, and all in all, we came up with the same findings, that we were getting a broad response against multiple variants, and we were getting a protective response in these different animal models. So very briefly, we took this to monkeys, and we found very potent responses, whether we gave a small dose or a large dose of the vaccine. That was several-fold higher, actually 1 log higher, 10-fold than people who had previously been infected. And we, were able to demonstrate this by different assays. And this translated into pretty potent and rapid protection as measured by virus in the lungs or virus in the upper airways. And one of the key points was that whether we looked by a live virus assay or a pseudovirus assay, and we looked across the different variants. So this is the original Washington 1, this is alpha, beta, gamma, delta. We didn't see much of a decrement in response. So much so that actually it wasn't statistically significant, the fold reduction in response. So we were still seeing very potent antibody responses. Well, that's all in the past. What about with Omicron? So we started to look at Omicron with BA1, the first subvariant, and we found, again, there wasn't really much of a decrement in response, and I'll show you that in the next slide as well. But we also started testing the antibody response, the binding antibody response against other viruses that are SARS-like but haven't made the leap into humans. We wanted to see how broad of a response we were getting. And this assay called biolayer interferometry, or we nickname it octet, we saw that it actually correlated well with neutralizing antibody response, such that a response of 0.5 nanometers corresponded around to about 1,000 in neutralizing antibody response. And so in these different types of SARS-like viruses, we were getting pretty good responses too. So we did a study in collaboration. We actually did 2 studies in collaboration with Dan Baruch at Harvard, and we gave 2 doses of our vaccine. Instead of at prime and 1 month, we gave it at prime and 2 months, 8 weeks, and then we challenged the monkeys 1 month after their second vaccination. And just before challenge, we see again their antibody responses against the original is pretty high, but it doesn't really go all that low for either Delta or Omicron. And if you challenge them with the BA1 Omicron, in the lower airways, in the lungs, we get very good protection compared to control animals that didn't receive the vaccine. In the upper airways, it's a little messier, but you still see a significant reduction in the virus in the upper airways. We've done the same study with a BA5 challenge. Both of these are under review. Manuscripts have been submitted. So now we get to the phase 1 clinical trial. We were really the first kind of broadly active, universal type platform to go into a clinical trial. We're still waiting on the ability to put all the data together to publish, but we're getting very close to that. We had a very dedicated group of volunteers very early on that stuck with us through the entire study. Even when we weren't able to enroll the full complement of individuals because we weren't able to find people who were naive to infection or vaccination. In the end, though, what we'll be looking at is not just the antibody response, binding and neutralizing antibodies against all the different variants starting from the original wild type Washington 1 strain all the way through BA.4/5 Omicron, but also we'll be looking at the antibody response against those other SARS-like viruses that only exist in bats or pangolins. And in collaboration with our great partners at Texas Biomed, we're going to be doing a study in which we're taking antibodies purified from these vaccinees, transferring them into hamsters, and then challenging them either with the Omicron variant or the original SARS-1 from 2002 to see really what kind of response we're getting, a protective response we're getting against viruses that go, beyond just SARS-2. What are the next steps? Well, I'm no longer there, but the next steps at the time that I left were to look at combinations of particles, also those mosaic particles where we stick on different types of spikes onto a single particle. We already saw early on from some mouse studies, if you combine particles, you get very good responses to both SARS-1 and SARS-2 that are in those particles. Combining platforms. So mRNA is not necessarily the panacea. Nanoparticles aren't the final solution either, the final answer. If you combine some of these platforms together, you actually may achieve some synergy. And we know that we're going to have to engage other parts of the immune response, the innate immune response, the T-cell response, not just antibodies, if we want to get broader and longer lasting immunity. And then what was controversial at the beginning of the pandemic, I think is not as controversial now, which is using human challenge studies to evaluate next generation vaccines. What I mean by that is actually what we do in animals, but doing in humans, taking people, vaccinating with a particular candidate, and then giving them virus. Now, we wouldn't give them SARS-2, we wouldn't give them SARS-1, but we might give them one of the milder human coronaviruses, like NL63, OC43, where we're sure that very healthy individuals will only get maybe a mild cold, but it'll give us a lot of information about the response to coronavirus vaccines in general. So finally, what does this look like if we were able to get a good vaccine that gives us broad immunity, broad protection, a universal vaccine in the clinic? How do we get that into a population? What does that look like? Well, first of all, you need the support, the political will, the funding. And fortunately, the U.S. government earlier this year really made a clarion call for developing those next generation vaccines. And they really are all, to this point, largely based on the nanoparticle approach, at least those that are the furthest forward. There are other strategies as well in terms of using other proteins, not just the spike or components of the spike. But the benefit of nanoparticles is that you're able to enable all these cells that make antibodies to co-locate with one another. And we know that increased antigen density engages multiple B-cell receptors at the same time, causes cross-signaling and potentiation of these B-cells to give a more mature and a more potent response. And we've seen that for nanoparticles, virus-like particles, for a whole range of pathogens. And so these are some of the other groups as well. We're certainly not the only ones who've been taking this approach, and they have other strategies for doing this. I should point out that, though, that two of these groups also have ferritin as the base of their approach as well. We were the first, but we were definitely not the only ones and not the last. And so ferritin definitely has some properties that makes it attractive as a next generation vaccine candidate. There's a lot of work going into trying to define what we mean when we say universal vaccines. So we have to define our nomenclature. And I think ultimately we need to take a phased approach. First and foremost, we need a pan-SARS-2 vaccine. We need a variant-proof vaccine. And then we can get to a pan-SARS virus vaccine. So all those different viruses that are currently in bats or pangolins or not even there yet, that still fall within that smaller cluster of SARS-like viruses. Then we can start talking about truly universal vaccines. I think it's a long ways off to try and cover the whole family of viruses, but maybe we can get to covering only those viruses that potentially cause severe disease. So MERS, SARS-1, SARS-2, they're all beta coronaviruses, all fall within that genus. And then we need to set the targets of what kind of vaccine we need, what kind of vaccine will we tolerate. We can't say 100% efficacy given only once for the rest of your life. That's not realistic. So we have to set what are the thresholds for what a good next generation vaccine will look like. And lots of different organizations are focused on this, developing target product profiles, preferred product characteristics, and roadmaps for what these vaccines will look like. They're all focusing on breadth and durability, but I think there needs to be even greater focus on the manufacturing properties of these different vaccine candidates. We need to make sure that the regulatory science is maturing along with the basic science and the clinical science of these vaccines. And we really need to ensure that the behavioral and social science follows along as well. Most of us developing the vaccines and involved in the product development pathway are not social scientists, we're not anthropologists. We need to engage those experts because the adage goes, vaccines don't matter, vaccinations matter. So what's the point of getting a wonderful vaccine if people aren't going to take it? So that needs to be really the end state of the product development cycle, not getting a efficacious, safe vaccine, but one that is acceptable and taken up by the people who need it most. We need to get ahead of the curve, literally ahead of the epidemic curve. And we need to do that with better surveillance, sequencing, synthetic biology, and bioinformatics. We need better platforms, prototypes, and we need to focus on broader categories of pathogens. We need better capabilities, competencies, and capacity in local research, manufacturing, and regulatory infrastructure at the places where these pathogens emerge. We cannot keep exporting all these capabilities during a pandemic. It needs to be there, ready to go. And it's not enough just to innovate. You have to incentivize that innovation, and you need to invest in that innovation for pre-positioned products. I hope this is the moment when we don't say, oh, it's over and let's go back to other things. I hope that we sort of work against human nature and still maintain the focus and the momentum on continuing the work for preparedness, engaging all the international stakeholders, and selling that mutual benefit to those stakeholders, that there are mutually derived priorities from continuing to do this work to develop these vaccines in advance of the pandemics. So this is the point I wanted to get to very briefly to opine but there are a lot of folks in here, I think, and I look forward to hearing your thoughts on this in future conversations of what is the U.S. military's role in medical research, biomedical research and development. For the longest time, it was really the only game in town. Before there was an NIH, before there was a CDC, it was the military, it was the Army, it was the Navy, it was the Air Force, doing public health, doing countermeasure development. But now there's so many different players involved. And the military recognizes that, and in some cases defers, in other cases tries to find niches. The model has always been identifying the threat, prioritizing the threat, developing the countermeasure, and then now partnering with the right collaborators and the right developers to get those products delivered to the military population and then by extension the global population. Is that still the model with all these other players involved? Is it more of a model of being a potentiator, being sort of an enabler of other organizations to do this? I don't know the answer, but it's something that I think needs to be thought about, and I know is being thought about quite a lot. So coming back to this was a golden age for vaccinology, but they hit the easy targets initially. So this here on the left is Albert Sabin, inventor of the oral polio vaccine. And this on the right is Maurice Hilleman, who led vaccines at Merck for decades. Both started their careers early on at the Walter Reed Army Institute of Research, or its forerunner, and were inspirations for me to coming to RARE in the first place. These are two of my heroes. The reason why I showed the picture of Hitler and Stalin in the first place was that it's always interesting to me that we know immediately the figures in history who have been responsible for the most deaths in history, but we really have no idea when we look at images of them like we do of those others, of the people who have been responsible for saving some of the most lives in history. And so, I don't think people would associate necessarily the US military with saving lives in this way, and what it has done for medical research and what it has done for vaccine research and development. And so I think recognizing and acknowledging that legacy in the evaluations going further for how the military is involved, how the global community is involved, I think these guys set a certain example. Clearly, the targets going forward are going to be a lot more difficult, but there was already a polio vaccine, right? When Sabin said, no, we got to get a better vaccine. There were a number of vaccines also for influenza, even hepatitis B. And these folks said, no, we got to do better. So even if we have a vaccine for something, which we do, it's not good enough if it's not getting to the whole world, if it's not getting to the levels of protection that we want it to. So we have to keep aspiring for better. The last thing I'll say is that this is basically another way of saying hindsight is 2020 when we say life can be understood backwards, but we always have to keep moving forward. So that's why we have to keep fighting the future wars. We can take the lessons from the past, but we have to make sure we apply them going forward. And so what is that path forward? I think there are multiple paths forward. There's not one single route. As I said, there's not one platform, there's not one antigen, there's not even one epitope. There are multiple ways, but it's going to take lots of coordinated investment and continued interest to be able to sustain those multiple paths. I'd like to finally just say this is just a small fraction of the folks that have been involved. And there are many others. This is actually one of our clinical slides. I didn't put all the preclinical slides on here as well. As I mentioned, Texas Biomed has been involved in a lot of the work that we've done. Southwest Research Institute has been a lot of work that we've done, other academic institutions. And then two of the folks here today were very instrumental in enabling a lot of what we did. What I realized during the pandemic is that leadership really matters a lot at the highest levels. And so I also want to thank General Talley, who's here today, and General Tahan, who's here today, for really letting us do the work as quickly as possible, and doing it in such a way that allowed us to collaborate across the agencies. That was what was so amazing about the federal response and the public-private partnership, was that folks from the government across agencies really tore down that red tape and let the science rip forward and get to products that could save people's lives as quickly as possible. I hope that's a model that can continue going forward. And I really want to thank, again, Dr. Hepburn for inviting me here today, and for all of you for taking an evening out of your time to spend with me. Thank you.
Dr. Mojrad, that was a tour de force, and I know we have a number of questions from the virtual team members, but I took away a couple key points that, one, and we learned this at the Uniformed Services University years ago, that emerging infectious diseases really are the Third Army, with incredible strategic, military, and global stability challenges. Dr. Mojrad underscored the importance of being proactive, the importance of synergy, military, academic, and industry, and having a global perspective, and having eternal vigilance in being as proactive as possible. And I know we have that ethos here in San Antonio, across our university and our institutions here in San Antonio, and we're honored to partner with great leaders. General Tan, thank you for your leadership. General Murray's leadership prior to that, and the brilliant scientists that you had at Walter Reed Army. They're incredible work. I don't know if it will ever be recognized, but we certainly do realize the importance that they had on the Operation Warp Speed over the last 2 years in protecting our nation, but also our allies around the world and the global humanity. So I salute you, Dr. Mojrad, and I salute you and wish you every success in Pfizer, because his new job will have global significance. So we have a tradition in the military, and many of you know that, of giving a coin. It's really not the value of the coin, but it's certainly the spirit that goes behind it. We wish you Godspeed, Kayvon.
Thank you so much.
All right, we also have a plaque for you.
Oh, thank you.
And we hope you'll have a place of honor in your new office in New York City.
Absolutely. Thank you so much.
All right, we'll do a little photo op here.
Okay. Thank you very much.
I want to respect everybody's time, but I do think, sure, there's some questions tonight. So, Andrew, were there any questions from the chat that we want to maybe, if you need to leave, then please, I want to respect your time. If you need to bail out, that's not a problem at all. But, Andrew, are there any questions you want to pop up? Yep, here.
I guess first we'll start off with Jeremy asks, how many spike proteins could you attach to a single ferritin structure? Is it 24?
It's 24 spike monomers or 8 spike trimers. And so that's that, you get 24 faces to work with, and that's it. But when you have triplets of a protein, it's 8 divided by 3, so you get 24 divided by 3, so you get 8 spike proteins.
Gotcha.
Let's shift over. And Dr. Gupta, you had a question, I think.
Very nice talk. And as a structural biologist, I really appreciate and thank you for illuminating the power of structural biology.
Absolutely.
My question is about SFNP, the ferritin-based vaccine. Did you engineer the iron binding site in the ferritin because it could potentially perturb a normal iron storage function in the cell?
Right. So we chose H. pylori ferritin intentionally so that there would be no cross-reactive immune responses to human ferritin. This has been tested a number of times in the context of other programs, particularly in the flu program, and we've been able to confirm it in our program that we get no cross-reactive responses, so that you're not causing any pathology and any kind of anemia in humans. But there is another concern here, which is when you have any kind of platform, whether it's a viral vector, a protein scaffold, you're going to develop antibody responses to that scaffold, to that vector. It's going to happen. So, and presumably the more you vaccinate with that same thing, you may get greater and greater antibody, you're getting a boosting effect. Will that abrogate the responses against what you're trying to focus the immune response to, which is the antigen? So far, at least from the flu program, we don't see that really having, but That's only been, 1 or 2 vaccinations, and it hasn't been carried out over a long period of time. We're looking at that as well in our clinical trials, not at liberty just to say yet what we've found, but from the flu program, on the short term, we don't see that much of an interference of the response against the antigen.
Thank you.
Dr. Shane Gunther, Colonel Shane Gunther.
That was a wonderful talk.
Thank you.
Thank you for pushing vaccinology forward for the military and for the world. I have one question. When we were conducting the trials at Burke Army Medical Center, first we had the Operation Warp Speed AstraZeneca trial, and then later on, a Pfizer Moderna came out. The primary concern, I think, for logistics was the cold chain problem, right? Yeah. We saw that on CNN, 60 Minutes and all.
Right.
With the ferritin molecule, will you have the cold chain concerns, or would it be just require refrigeration, or?
Every platform has its advantages and its drawbacks. What's so great about mRNA is the ability to scale up quickly and the ability to turn on a dime and insert a sequence, and now you have a new vaccine. Protein takes a lot longer in terms of developing the stable cell line to express your protein at certain levels and characterizing it and making sure what you're putting into people is exactly what you want to be putting into them. The benefit of protein as compared to mRNA is that protein vaccines are very stable. They're very thermal stable. They're very stable and resistant to degradation from freeze-thaw cycles. Ferritin is actually no different, and in many ways, ordered, structured particles, whether they be protein nanoparticles or virus-like particles, have a very good stability profile. We've done some of that stability testing on our drug product in preclinical studies and then on the clinical material as well. And we'll include that in our publication too, but suffice it to say that nanoparticles, being in the broader category of proteins, don't require that same kind of cold chain that mRNAs. But there's a lot of advances also being made in improving that stability profile of mRNA. There's still a ways to go, but it's actually improved even from the earlier days of the pandemic.
I want to respect everybody's time tonight, but I'd be remiss in not recognizing Major General Talley. Good to see you, sir, and your beautiful wife there, and thanks for coming down. I apologize for not introducing you earlier. General Talley led the research command up at Fort Detrick there, and now you're here in San Antonio. It's good to have you here in the neighborhood, so that's great. Look, it's been a great evening. Kayvon, it was so much, and we wish you and your family Godspeed. And for those that are on the virtual net, thanks for joining us tonight. And we're going to record this, so please share the recording, and the link for it, we'll share that with all of you so you can get it out to your colleagues that weren't able to be here tonight. But we wish you well, and for the future doctors and researchers that are in the audience, we have many of them. We know you've got worthy work ahead of you, and we wish you all Godspeed as well. Have a great evening. Thank you so much.
Thank you very much.